Hello everyone, I am Adrian Moore, and today I'm going to talk to you about some of the research I've been doing as a postdoc at the University of Minnesota. The overall project is using a life history guild approach for freshwater mussel risk assessment, and I'm going to talk to you today about a small piece of that where we use dynamic energy budget modeling and look at individual level responses. As many of you know, U.S. freshwater mussel species are highly diverse, but also in decline. About 90 out of 300 species in the U.S. are listed as threatened or endangered under the Federal Endangered Species Act, and these species exhibit a wide range of life history traits. I particularly like this chart from North American Freshwater Mussels, 2012, that shows the growth curves of a lot of different species. And we can see there's some species like Leptodia fragilis up here that has quite a short lifespan, but grows to be quite large in that time, compared to other species like Quadrilla pustulosa, which live to be much older, but don't get nearly as big in that time. Pesticide risk assessment for freshwater mussels presents a lot of challenges. To start, there are a lot of species to consider. And laboratory testing is difficult or impossible for a lot of these species, especially those listed ones with small population sizes. The eastern oyster, Chrysostria virginica, serves as the surrogate for toxicity testing for pesticide registration, but we don't really know how well toxicity in the eastern oyster relates to that of freshwater mussels. Additionally, we have a lot of data gaps in the important life history statistics that we might use to understand how these individual level impacts translate to population level effects, which is where our conservation goals are. Effects modeling can allow us to overcome some of these issues by extrapolating toxicity among species and allowing us to translate from individuals to populations. The effects modeling I'm going to be talking about today is the dynamic energy budget theory. This is a generic metabolic theory that describes the energy allocation in an individual. So you can see in this schematic over here on the right, energy is assimilated from the food the individual eats into a reserve. Energy from that reserve is then mobilized to the somatic branch or the maturity and reproduction branch. Within each branch, a certain amount of energy is used for maintenance, and then any energy left over can be used for growth or maturation or production of gametes. And there are a lot of advantages to dynamic energy budget theory. To start, it's generic, which means that when we want to look at multiple species, only the parameters change. The base model itself does not change. The European Food Safety Authority has also identified DEB as a viable method for pesticide risk assessment. It's been widely used with bivalves, though mostly marine up to this point. And it can be parameterized with limited life history data, which allows us to get over the fact that we don't always know all the life history traits of the species we're interested in. And finally, it can be used to quantify responses to stressors using these defined physiological modes of action, or PMOAs. We have 4 that we generally consider. Assimilation refers to a decrease in the amount of energy that comes from food. Maintenance refers to an increase in the cost, the energetic cost of maintaining the individual. Growth means that it takes more energy to increase growth. And then reproduction is a reduction in the amount of eggs that can be produced from the energy available. So the objectives of this project are to compare the effects of hypothetical stressors on individual level endpoints of freshwater mussels within different life history guilds. Many of you will be familiar with the life history strategies defined by Wendell Haig in North American Freshwater Mussels 2012, and we're going to use those same guilds to separate our mussels into different life history guilds. And so the first goal was to compare the responses, length and fecundity of mussels within these different life history guilds to hypothetical stressors. And our second goal was to compare the freshwater mussel responses to that of the eastern oyster, which is our toxicity testing surrogate. Our approach was to select one listed and one non-listed freshwater mussel species from each life history guild. We did this using our own database of life history trait data and replicating the methods of Haig in his original approach. So we use the same traits, the same methods, but different data to pick our species. And we ended up with these species over here in this table on the right. Mucket and southern clubshell are equilibrium species. Butterfly mussel and fine line pocketbook are the periodic species. Fawn's foot is our opportunistic species. There were no listed species in the opportunistic category, so we don't have a listed species there. And then we lastly have the eastern oyster, which will serve as our surrogate. I wanted to quickly show you some of the trait data for the species we'll be working with. These are growth curves here on the left, so age on the x-axis, length on the y. Our eastern oyster is here in black. You can see that it has a significantly shorter lifespan than many of our mussel species, but grows to be quite large compared to some of our mussel species, which live longer but don't get to be nearly as large. Our opportunistic species is here in green. You can see that it does not grow very large nor has a very long lifespan. On the right, we have fecundity, annual fecundity on a log scale. Again, you can see the eastern oyster has quite high fecundity compared to the freshwater mussels. But among the mussels, both our mucket and our opportunistic species, Chinchillida naciformis, have quite high fecundity compared to the other three mussel species. Once we had our species, the next step was to parameterize them in a DEB model. We did this using a service called DEBer. And this process uses known life history traits as well as Bayesian analysis to generate an ensemble of potential parameter estimates for each species. And this ensemble of parameters allows us to capture some of the uncertainty in our parameterization process. Using these ensembles, we ran 100 replicates of each at a 0% stress to serve as a baseline, and then 50% stress for all 4 PMOAs, and the stress was applied over the entire life cycle. So now I'm going to show you the results going through each PMOA one at a time. We have our species on the x-axis, so you'll notice our eastern oyster is here under Seaver. Our opportunistic species here, Chinchillida naciformis, is here on the end, and then our equilibrium and periodic are the other 4 positions. On the x-axis, we have our ultimate length, or maximum size, and this is going to be percent difference relative to the baseline. On the right graph, this is annual fecundity, also relative to the baseline. So for our assimilation PMOA, which is a reduction in the amount of energy the individual can take up, we see that for all 6 species, there is about a 50% reduction in the maximum size of the individual. When we look at fecundity, we see a much greater effect, around 90 to 95% for all of our species. And then our opportunistic species, we see a nearly 100% effect in many of the replicates. So it's affected a little more strongly than the others. Looking at growth, again, we see that most of the species respond very similarly with almost 0 impact to their ultimate size. Our eastern oyster, we see there's slightly more effect with about a 7% decrease in its maximum size. Over on the fecundity side, we see that the impact again is a bit greater for all the species, but especially for the eastern oyster, where there's about a 15% decrease in its total fecundity. For the maintenance PMOA, meaning more energy is needed for the regulation of homeostasis, we see that there is, even at a 50% stress, there's about a 30% decrease in the maximum size of the individual. For fecundity, we see that the eastern oyster is slightly less affected than the mussels, coming in at about a 52% decrease compared to closer to 60 for most of our mussel species. And again, our opportunistic is the fecundity is more greatly increased, coming in at an 80 to 100% effect on reproduction for this species. I'm not going to cover the reproduction slide because it's quite boring. There's no effect on growth, and fecundity is always cut exactly in half. So didn't really need a graphical representation. But to summarize these results, what we saw was that under most of the PMOAs, the impacts on length were very similar among the freshwater mussel species, but for fecundity, we saw some variation. And in particular, our opportunistic Trinsella denasiformis, the fecundity was impacted more by stress on maintenance than other species were. The response of the oyster was also similar in respect to length, but again, the fecundity of the oyster was sometimes more sensitive, was sometimes less sensitive, so it didn't always align perfectly with our freshwater mussel species. But overall, we did always see that fecundity was more strongly impacted than ultimate length was. To relate this back to our original objectives, to compare among mussel species with diverse life history strategies, what we saw was that the periodic and equilibrium species largely responded the same for both endpoints and for all 4 PMOAs. But our opportunistic species, the fecundity was sometimes more strongly impacted. And to compare to the freshwater mussels to the oyster, we saw that the relative effects on the oyster was again similar for length, but not always the same for fecundity. And sometimes those responses varied from the freshwater mussel responses. So I want to point out some of the limitations with the data and approach that I've discussed here. These endpoints are only ultimate values, so the maximum size and the annual reproduction at the end of its life cycle. And that could look very different if we were to look at the endpoints differently, say cumulative reproduction over the full life cycle. We also didn't address all the potential endpoints. I particularly think that age at maturity may be consequential for freshwater mussels and could be impacted by these PMOAs. It also doesn't account for some of the complexities of the freshwater mussel life cycle, such as brooding and metamorphosis. We know that gamete production does not necessarily translate directly to viable glaucidia, and we especially know that the number of glaucidia doesn't directly translate to juvenile recruitment. And so we need to account for that in other ways. And then lastly, these PMOAs are hypothetical stressors, and while they can be linked to actual toxicity scenarios, we need a lot of data to do that, including reproductive data, which we can't get with freshwater mussels at this point. And so to take this back to our larger project, I want to discuss what this means for risk assessment. What I think it tells us is that extrapolation of toxicity test results from one species to another can be appropriate, and so it's possible that we can use the eastern oyster data that we have a lot of to substitute for freshwater mussel toxicity test data that we are missing. But what we do know is that individual level responses do not translate directly to population level responses. And my lab mate, Chiara Cola, has a really nice demonstration of this with trout species. So our next step is going to be a population model. So we're going to integrate this DEB model that I've presented on today with different mussel traits like its reproductive phenology and host fish, as well as environmental conditions like food and temperature. And we're going to use this to predict how dynamics at higher levels of biological organization, like the population, emerge from these individual level processes. Finally, I'd like to acknowledge some of the people who've helped me with this work, and then my funding sources, Syngenta Crop Protection and the University of Minnesota. Thank you.